All. This is Dr. Mubin Sayed from drbeen.com. Welcome to one more show. Today, I have a beautiful story with great graphics for you. And today's talk has the world's very first discovery of three-dimensional pattern recognition receptors in the bacteria that bacteria use to kill or to die when infected with bacteriophage. This particular study and the when I was reading it and then drawing it, it has made me so excited. I'm sure that not everyone would uh, be as excited as I am, but I'm a mechanisms guy and I love these mechanisms. Let's look at it. This is the world's first ever discovery. This would change our future of medicine for antivirals. This would actually change our understanding of bacteria. This would change our understanding of our own body as well. This is such a monumental discovery. So let's look at it. So, first of all, these are our gifts for humanity. They're continuing. Then, this is drbeen.com. There is a description. There is a link in the description of this video with, I stopped to say, just $67 one-time fee to get access to 900 videos like these videos that we'll do today. So take advantage of that. And here is the researcher, Jonathan Kagan from the Boston Children's Hospital. So love his work and salute to him for what he and his team did. This is an article about it. This is the actual study. Then some more references as well that may be of interest. And these references are present in the description of this video. With this, let's start. So today, get ready for a tiny story. Imagine somewhere some bacteria are sitting and playing. Maybe they're at a beach and they're playing beach volleyball with bacteria. Maybe they're using one of the coccus. Coccus means rounded bacteria. So maybe they're using one of the cocci and using that to play. So anyways, they're playing. You can see some bacteria here. This is a streptococcus. These are staphylococci. Seems to be an E. coli. And this seems to be some clostridium of some sort. So they're having fun. Then they are so scared now. They're totally scared and they are all nervous and they're crying. Why? What happened? What happened? Do you know the pandemic that happened to humans? Infections can happen to bacteria as well. So these bacteria are all nervous because a bacteriophage has arrived, or not just one. Millions of bacteriophages are here. These bacteriophage, or simply called phages, are the viruses that do not infect humans. Instead, they infect bacteria. And something fascinating, again, the healthcare workers or professionals or students, healthcare students or molecular biology students, they would know this already. But do you know that these phages, these viruses, are actually specific for various genera or strains or variants, species of bacteria as well? Just like we have human viruses, these are bacteriophages are bacteria viruses, but they can be further classified according to the type of bacteria they infect. And this is why bacteriophages can actually be detected in wastewater to try to figure out what kind of bacterial disease is present in the community. Why? Because in the wastewater, bacteria may become destroyed. However, these little pieces of this little viruses can just keep surviving. And so you can use them as a surrogate to understand in what community, what kind of bacteria may be present because their infecting phages are present in the water, wastewater. Then, if you see here, there are two phages actually. This is why these little bacteria were all kind of all upset and nervous. Why? What's happening? Phages attack a bacteria. They have specific structure. Imagine these phages are like drones, those flying drones that we have, which children or professionals fly. Imagine these are drones. In their little head, they have actually head and tail and legs. So in their little head, they pack a brain, small genomic material, small DNA material in them. You can actually pack anything in them, and these little drones, vehicles, would just take that little genomic material to the next bacteria and inject it in there. This is their function. This little machine does that. It doesn't care. Neither does it have a brain to figure it out. It doesn't care what is packed in it. It will just take it. It is a perfect pathogen inside our body to transport poisons and toxic substances equal to drugs in the human society, because these guys have no brain. They'll just pack something and they'll fly to the other pathogen. Okay. Because of this, these bacteriophage do something very interesting. They can actually, and so if you see the second bacteriophage, it has taken its genetic material and injected that in this bacteria. Now, this genetic material would hijack the bacterial machinery, just like the viruses hijack our cell machinery. And then this RNA or DNA would help make more bacteriophages. However, a bacteriophage, this little virus of the bacteria, is responsible for carrying lot of toxins. The bacteria have toxins, right? So when bacteria are present in us, many bacteria produce toxins. You know that bacteria acquire those toxins from the DNA of these bacteriophages. So the recipe for the poison that a bacteria produces, bacteria acquires that recipe from a bacteriophage. This is like when a baby bacteria is born, it has to go find out a bacteriophage and say, can you please give me some poison powers? And then the bacteriophage would inject the genetic material to make toxins, and then the bacteria would start making those toxins. Many bacteria that are toxin producing, if they do not encounter a bacteriophage, these bacteria stay non-toxin producing, non-lethal, non-dangerous. So that is a function of bacteriophages. Another function of the bacteriophage is accidental movement of bacterial DNA. So when the bacteriophages are now going to be made inside the bacteria, they sometimes accidentally pick up pieces of DNA of the bacteria and take them to the other bacteria in the vicinity and inject those DNAs in them. Do you know this is such an important thing for the microbiome of our gut? Our microbiota is filled with the good bacteria and bad bacteria. And sometimes these bacteriophages can pick up genes from good bacteria and bring them to the bad bacteria and convert the bad bacteria. So these bacteriophages are important to keep a healthy, correctly functioning, good microbiota for us. And for the healthcare workers, professionals, students, I think you already know when a bacteriophage moves the genetic material from one bacteria to the next. This is called transduction. Transductions are of two types, generalized transduction and specialized transduction. In generalized transduction, bacteriophage just accidentally picks up some piece. It didn't want to pick it up, but it picked up some DNA from a bacteria and brought it to another bacteria. That is called generalized transduction. It is really movement of a gene from one bacteria to the other. This is like if somebody comes in and picks up a piece of a brain from a human being and brings it to another person and installs it over there. That is exactly what's going on if bacteria were human. Now, the specialized transduction is when bacteriophages molecular machineries intentionally pick up pieces of bacterial DNA. When they are making their own DNA copies, they also pick up parts of the bacterial DNA as well and then take them to the other bacteria. This is called specialized transduction. So without bacteriophages, one will have bad bacteria grow all over the place, even good. Number two, keeping the bacteria in the good way, on the good side, will become difficult. Okay, so that is the bacteriophage. Now, how does the bacteria that is infected by a bacteriophage, what happens in it? This is very similar to our cells as well. So now this little red area, red thing, is the bacteriophage's genetic material. It hijacks the bacteria's machinery, hijacks its ribosomes, and starts making its proteins. These are the proteins. The ribosome, sorry, the genetic material is passing through the ribosomes. Multiple ribosomes can attach to one string of genetic material, and they can all quack, 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 work on it to make proteins. And here the proteins are coming out. You can tell how excited I am about these mechanisms. And this is the very first time ever we are knowing in the world, the very first time ever. Okay, so these proteins are formed. These proteins are the proteins that will make new bacteriophages. Plus, as part of preparing for the daughter bacteriophages, the genetic material will be copied as well. Then what happens? We are still inside the bacteria. Imagine this is this little guy standing here. This is an enzyme that is made by the DNA or the genetic material of the bacteriophage. So this is a viral enzyme. Just remember, like COVID has its own enzymes as well that are formed in our cells and they function to make more SARS-CoV-2s. Similarly, this is a viral enzyme. This enzyme's function, and why am I talking about this enzyme? Because this research used this enzyme. That is why. This enzyme, its function is to spool up. This is the genetic material for the new bacteriophages. We need to spool up or kind of pack up this little DNA string. It is a long string, and we have to kind of wrap it and pack it and assemble it into a small ball like structure, and then insert it in these new heads that are being formed for the bacteriophages. So this enzyme picks up the copy of the genetic material, spools it up into small balls or structures, 3D structures, and then pushes that in the head of the bacteriophage. Then the legs and tails will be formed, and then they all will be assembled, and new bacteriophages will be formed. Good. This is still happening inside the bacteria. Now, this is the key point for this study. The researchers found out that these DNA spoolers, let's call them, or DNA packaging enzymes more accurately, these DNA packaging enzymes can be different. Their genetic material, of course, there are going to be genes that, when translated, first transcribed, then translated, they will make these proteins. These proteins are DNA packaging proteins. And the researchers found that the genes in various bacteriophages for DNA packing proteins were different. Here, this is one gene in one bacteriophage. This is a gene with some mutations in it, just like SARS-CoV-2 is doing mutations. This gene has some mutations. It is still making the packaging DNA packaging enzyme, but the gene is slightly different, mutated. This gene is even more different. This gene is different as well. So if you see, they're all slightly different proteins, although they still do the same function of wrapping up the DNA and putting it in the head of the bacteriophage. Why did I talk about this particular thing? Because imagine you and I are tasked to identify this enzyme's presence. And what we are doing is we said, you know what, let's take a microscope and look at these DNAs all the time. But it turns out that DNA has so much mutations that we cannot really correctly identify. We identify one kind of DNA, and the next bacteriophage has a mutation. And now our pattern recognition is not working. So now think about it for a second. That is a problem for this bacteria as well. How would the bacteria fight with this virus? Because it has to recognize somehow these genetic pieces. But these genetic pieces have so much diversity that a bacteria itself is a single cell or lower than a cell, right? It's not even a cell, correct? How can that have innate arm and acquired arm and neutrophils and the macrophages and the mast cells and the T cells and B cells and livers? And it doesn't have all of that. So bacteria has a problem. The problem is, how do I identify these viruses to respond to them when I become infected? So bacteria does something really fun. This is the first time ever discovery. So before we go to what it does, important thing to notice, bacteria commits suicide once it knows I am infected. It doesn't have the immune system privileges like us, a polished, huge organ that helps us. Bacteria can just identify a virus. Even just identifying a virus is a big deal for this lower than a cell organism. Once it identifies that, all right, I have a virus, it would just cause apoptosis and die. Why would dying be better for this bacteria? Because this bacteria would die and the colony around it will be safe. Otherwise, this bacteria would not die and the virus would keep replicating in it, and the millions of viruses would come out and attack the colony next door. So, bacteria just dies so that all the actions inside are stopped. Now, we are still back to this at why and how does it know? So, let's answer that question. Here is this is the discovery, first ever in the whole field of medicine and molecular biology. This is the first ever discovery. Here is what they found. They found that bacteria have a protein called AVS protein. They named it AVS. These AVS proteins are not looking at genetic material, or they are not looking at tiny protein pieces of the bacterial enzymes. Instead, they are looking at the whole protein structure. Imagine as if you have a hand and you put that hand, so let's say this is the cup in my hand, you put your hand on the cup and feel the whole structure, three-dimensional structure. So imagine you have a pattern that can fit like a glove on top of the three-dimensional viral proteins. Now check this out. Virus could decide, the bacteriophages could decide that my protein is going to be slightly different. Fine. These are all different. But because they do the same function, they still have to have a structure that allows that function. The form follows the function, or function follows the form, right? So the form, eventual 3D shape is the same, almost the same, even when their genetic materials are different, because those genes eventually are making proteins to do a specific function. And so when the protein finally coils into a 3D shape to make that function, the protein is almost always very similar. So the researchers actually made mutations to the protein intentionally and then used AVS to see, can the bacterial AVS still recognize even with the mutations in the DNA? And yes, as long as the protein that was formed was mostly similar to perform its function, it was detected in this with this three-dimensional pattern recognition system. This is like if you close your eyes and with your hands you feel the three-dimensional shape of something and say, well, this is this thing. That's what this bacterial sensor does. It is as good as our complete innate arm. Okay, so now the problem is on the bacteriophage. How does bacteriophage try to escape its proteins from getting recognized? Remember, the bacteria will sense it, recognize it. Once the recognition occurs, that protein, this little red hand that I've made, these are bacterial proteins, AVS proteins, that are able to kind of feel these proteins from bacteriophage. These proteins, the bacterial proteins, they have a little end on them. Proteins have carboxy and nitro end, or N end, amino end. So their amino end, imagine, has a little grenade attached with it. So as soon as this glove fits, this grenade goes off and bacteria is destroyed. Actually, our innate arm also have such receptors, but not, we never knew they are 3D receptors. We just knew they are pattern recognition receptor, and they are partial pattern recognizers. This is a complete 3D pattern recognition. This is just a marvelous thing that they found. So now, let's say the bacteriophage says, you know what, I need to change my DNA so that the enzymes that I make, their shape is going to be different, so that this stupid AVS sensor protein will not be able to sense it. And that way I can escape the bacterial destruction. And bacteria would not sense me, bacteria would not do apoptosis. I would continue to make more bacteriophages in it. My factory would stay intact, and there I am. So they decided to change their shape. Now let's see. Here is the shape they changed to. Can you see the problem? They have definitely changed their shape, mutated enough that the sensor will not recognize them anymore. They have escaped immunity. They have escaped the innate arm of the immunity in the bacteria. However, I think you can figure out that these are not going to function for what they are supposed to do. They're not going to be packaging DNA and putting that in the bacteriophage's head. There is the shape is all totally a mush. This looks like a little llama without legs to me, but anyways, the shape is different. That means if bacteriophage tries to escape immunity and it changes its enzyme, then those enzymes become functionally useless. And so that bacteriophage is not going to go anywhere. If it does not change the enzymes, then the bacterial sensor, AVS sensors, would detect it and cause apoptosis of the bacteria, and replication stops. What a beautiful way. What a beautiful innate arm just with one protein. So here, if the bacteriophage decides to change the protein, then what would happen is the protein will become useless. It would not be able to hurt the bacteria. So here I made a little cartoon. This is a bacteriophage with the mutated proteins, which are useless. So bacteria are coming over and saying, sir, accept this Darwin Award from the culture of bacteria and partners. And here is the award this bacteriophage is getting for the mutation. I I spent some time drawing these for you today. Okay, so the AVS protein recognized the DNA packaging proteins of human herpesviruses as well. This is even bigger news. So far we are talking about the bacteriophage that is infecting a bacteria, and bacteria is trying to sense the bacteriophage and respond, correct? and we found a new protein there. Then the researchers took that protein, right? At the end of the day, we are selfish. It's all about humans. So we took that protein, AVS, and used it on human herpesvirus, so a human virus, and try to understand if it can bind with the human, it can recognize the DNA packager of the human herpesvirus. And guess what? It did bind with it. It was weaker as compared to it would bind with the bacterial packagers, but it was able to bind with the herpesvirus's DNA packagers. Can you imagine the future of antiviral therapies? So that was exciting. Then, final thought. You might say, some of you might say, that wait, our innate arm does this as well. We also have pattern recognition receptors, and we've been talking about it for 2 years. So, yes, our macrophages, dendritic cells, neutrophils, many other cells, especially on the innate arm side, they have pattern recognition receptors. What these are, there are two types of them. One that are on the outside. Imagine your arm dangling outside of your body, and with your hands, you can feel various things in the environment. So, this is a receptor or a sensor popped out in the environment. And this is called a toll like receptor. And it can detect various patterns on bacteria and viruses and fungi and so on, broken cells as well. We call them PAMPs and DAMPs. Pathogen associated molecular patterns are called PAMPs, or DAMPs are damage associated molecular patterns, that are the patterns found in our damaged cells. However, these toll-like receptors are not known to fit a protein with a 3D structure around it so that they can still feel the functionality of the protein instead of a piece of it. There is a huge difference between TLR and AVS of bacteria. Bacterial AVS can close its eyes and feel the 3D structure of the protein and tell what it is. RTLR cannot do that. It can only sense a part. It can just put its hand on some surface and just say, well, I feel it is this surface, this pathogen surface. If the pathogen changes that surface, then the innate arm will be defeated. Now, inside the cells, dendritic cells or macrophages, mast cells, inside of them, there are also pattern recognition receptors, and this is in many cells. And what are these? Sometimes, remember, the dendritic cells, for example, will do phagocytosis, and they'll bring a pathogen, they'll arrest it and bring it in and then break it up and offer it to the systems inside to explore what it is. Or it is possible that a pathogen has actually infected the cell, and the pathogen, virus, bacteria, the patterns are present inside. So inside the cell, there are what is called NLRs. NLRs are nucleotide binding oligomerization domain like receptors. What does that mean? When we have little patterns, not a 3D structure, when we have little patterns of some bacteria or the virus, then these NLRs can bind to various structures or surfaces of it and detect that pattern, PAMPs and DAMPs. And based on that, they can then activate within the cell, they can activate something which is either apoptosis or caspase's activity and so on. The eventual result is a cell can die. So this recognizing the pattern and dying is very similar to bacteria recognizing the protein and dying, but bacteria is doing something else. Now, if I bring myself in front of you, bacteria is looking at the whole 3D structure just with one protein, AVS. AVSs are of multiple types too, but anyways, and then just feeling it. and saying, well, this is a DNA packager of a virus. I'm going to die. Our pattern recognitions do not do this. We do not take care of the whole 3D structure. We just look at one tiny part, maybe this much of a handle, and say, well, it seems like this must be a little mug. But we are not able to look at the whole 3D structure. So then, in therapy, how can this help? In therapy, this would help when we will form AVS or find AVSs that can recognize various bacteria and viruses and their enzymes, the whole structure of them, and then use them as therapeutics. Beautiful, beautiful. So this is the discussion. Thank you very much for listening to my, let me see if I have any other. So that's it. Listening to my little exciting news. And thanks to Kamran Jamil on Patreon, who sent me this link. So Kamran, thank you very much for this contribution. This is a great, great discovery, and I totally loved what it has in it. And now for the cool beans, please like, subscribe, and share. I do a lot of hard work to bring these news for you. So you could do a small thing and you can like, subscribe, and share. If you would like to support this work, there are links in the description. You can become a patron. You can just have $5 a month, and that is good enough. You can be a patron, or you can use PayPal, or you can buy me a coffee. You can become part of Substack. You can even become a member on Drbeen. The link is in the description, or become a member of YouTube. So there are so many options to become participant in this effort, educational effort. So with this, thank you very much. Stay safe, happy, and healthy. I would see you tomorrow.